Alright, so now we're going to start with the next topic, which is amino acids. Amino acids are the building block of proteins. Proteins, as we know, they carry out a lot of different functions in the bodies of living things, and amino acids are the constituents of proteins. Amino acids are not only the constituents of proteins, but they're also important for several other functions taking place in the cells. We will look at some other roles of amino acids at the end of this session. Here in this diagram, you can see the proteins, a few of the proteins here. For example, this is a firefly. The firefly, it produces light. That is because of an enzyme called as luciferase. Enzymes are also protein in nature. Here you see a picture of red blood cells. The red blood cells contain this protein called hemoglobin. Hemoglobin is important for the transport of gases. It is not only needed for the transport of oxygen, but also it is important for the transport of carbon dioxide. There's a rhino you see here. So the horn of the rhino, the hooves of the animal, and the hair on the skin are all made up of different types of proteins. Similarly, the claws, nails, they all are made up of structural proteins. So proteins are important for various functions in the bodies. For example, an enzyme shown here, a transport protein, as well as the structural protein. This is a generalized representation showing the structure of an amino acid. As the name show that it is an amino, so that means it contains an amino group on it. It is an acid. The acid group present is a carboxyl acid. So every amino acid contains these two essential groups on it, one amino group and a carboxyl group. These groups are attached to a central carbon atom. This carbon atom, in most cases, is called as the alpha carbon. To this carbon, a hydrogen is also attached. This is sometimes called as the alpha hydrogen. All amino acids share this same basic structure. There's an alpha carbon to which an amino group and a carboxyl group are attached, and there is a alpha hydrogen. This R group shown here is called as the side chain of the amino acid. The 20 amino acids, they all contain different types of side chains. We will look at the structure of those 20 standard amino acids later on during this session. These groups, the amino group and the carboxyl group, they contain dissociable protons on them. This is the carboxyl group. This carboxyl group can release this proton and can become a carboxylate group, shown here. Similarly, this is the amino group represented here. The amino group, the nitrogen atom of amino group, contains lone pairs on it. And by means of those lone pairs, it can take up a proton and can become protonated. So this is represented here. Amino acids, they, when present in the solution form, they are present in their ionized forms. The carboxyl groups, they deprotonate, and the amino groups, they are protonated. This is called as the zwitterionic form of the amino acid. So the amino acids, they have charges on them, and because of the presence of these charges, the proteins are also charged. This is showing the alpha amino group and the alpha carboxyl group, which are charged, but several of the groups present on the side chain, they are also charged. So these charges present on the side chains, they give a particular charge to the protein. Depending upon the position of the groups in space, like the way we have seen for carbohydrates, that there are two possibilities across the chiral carbon. They are regarded as the L form or the D form. So we have seen that if the hydroxyl group is projected on the left side of the chiral carbon, that is L, and if it is projecting towards the right side of the chiral carbon, that is called as the D form of glyceraldehyde. Similarly, the amino acids also have such type of stereoisomers. So if the amino group of an amino acid is projecting on the left side, that is called as the L form of the amino acid. And if it is projecting on the right side of this chiral carbon, then it is called as the D form of amino acids. Glycine is the only amino acid which does not contain a chiral carbon in it. All other amino acids, they contain this chiral carbon in them, and D and L isomers are possible for them. However, in proteins, the L amino acids are the prevalent ones which are found. D amino acids are only found in some prokaryotes and to a limited extent. Virtually all proteins, they're made up of L amino acids. This is a table from Voigt and Voigt, and this table shows some of the important amino acids. We're now going to look at the structure of these amino acids one by one. So the first one shown here is glycine. Glycine, it is abbreviated by this three-letter code GLY, and the single letter code is G. Glycine is the simplest amino acid. It contains a hydrogen atom as its side chain, or the so-called R group. So here you can see that this carbon is not a chiral carbon. So glycine is the only amino acid which is not chiral. With all other amino acids, they contain a chiral carbon in them. So glycine is the simplest amino acids, having a hydrogen atom as its side chain. The next amino acid is alanine, which is abbreviated as A. Alanine contains a methyl group on it. Well, it's a tricky job to remember the structure of the amino acids, but indeed it is important for you to remember these structures. So I'm going to talk about a few important things pertaining to the structure of these amino acids. You can first try to copy them from the book, and then later you can try to reproduce these structures based upon your memory. So it will take a little bit of a practice, maybe like about 2-3 times. If you would draw these structures, you would be able to remember them properly. So if you know about the basic skeleton of amino acids, that is, every amino acid contains a carboxyl group and amino group which are linked to an alpha carbon. So it would be very easy to remember the structure of glycine, because glycine contains a hydrogen atom as its side chain. And then it would be easy to remember the structure of alanine as well, because alanine contains a methyl group as the side chain. The next amino acid is valine. The amino acid valine contains propane. It contains a 3-carbon chain as its side chain. So you can see that this is 1, 2, and 3. So it's a propane chain as the R group of amino acid valine. The next one is leucine. Valine contains 3-carbon chain as its side chain, whereas leucine contains a butyl chain 1, 2, 3, and 4. So it contains 4 carbons, a butyl chain as its side chain. The next one is isoleucine. Isoleucine is an isomer of leucine. Isoleucine also contains 4 carbons as its side chain 1, 2, 3, and 4. So it is an isomer of leucine. So if you switch place of this methyl group and you place it on this carbon, so leucine will become isoleucine. The next one in the series is methionine. Methionine is the first amino acid during protein synthesis. You would be knowing about the start codon ATG, which on the messenger RNA is AUG. So the start codon AUG, it codes for the amino acid methionine. So methionine is the first amino acid to be incorporated during protein synthesis. So this is the side chain of methionine shown here, CH2, CH2, S, CH3. So the amino acid methionine is one of the amino acids which contain a sulfur in it. The other amino acid containing sulfur in it is cysteine. We will look at the structure of cysteine in the coming slides. The next one is proline. Proline is different from other amino acids in the sense that it contains a 3-carbon side chain. It's a propyl chain, but the 3-carbon chain is linked on one side to the alpha carbon, and it is linked to the amino group, the alpha amino group on the other side. So you see this is the alpha carbon here, this is the carboxyl group, the amino group, and the R group, the side chain, is linked to this alpha carbon. So 1, 2, and 3. So it's a propyl chain which is linked to the alpha carbon. But on the other side, the propyl chain is linked to the amino group here. So the side chain forms a cyclic structure with the alpha carbon and the alpha amino group of this amino acids. The next one is phenylalanine. You would remember from the previous structure, alanine. Alanine is an amino acid that contains a methyl group on it. So this is a basic skeleton of an amino acid, and if you put a methyl group here, that will become alanine. To alanine, if you add a benzene, this phenyl group, when it is linked to alanine, it becomes the amino acid phenylalanine. The next one is tryptophan. Tryptophan amino acid, it contains the indole group attached to this methylene group here. So again, if it is a CH3, then this would be alanine. If you would remember the structure of alanine, just put the indole group next to this methylene group, and this will become amino acid tryptophan. All these amino acids represented on this slide, their side chains are nonpolar. Okay, so now we next look at the amino acids where the side chains, they are polar, but they do not contain charges on them. So the first one here is serine. Again, if you would remember the structure of alanine, that this is the basic skeleton of an amino acid to which a methyl group is attached. If a methyl is attached, that would be alanine. So you just take one of the hydrogens of alanine away and put a hydroxyl group here. So that would become amino acid serine. Serine is a polar amino acid and it contains a hydroxyl group on it. The next one is amino acid threonine. Threonine contains a 2-carbon side chain on which one of the carbons it contains a hydroxyl group. So it is CHOH CH3 is a 2-carbon side chain with a hydroxyl group on it. So this is also a polar side group. Tyrosine is an aromatic amino acid and it also contains a hydroxyl group on it. Again, if we would look at the structure till here, this is the basic skeleton of amino acid, a methyl group. That if there is a methyl group, that would be alanine. So if you have alanine and you put a benzene here, so that will become phenylalanine. Onto this phenylalanine, you attach a hydroxyl group, so this will become the amino acid tyrosine. So amino acid tyrosine, threonine, and serine. These are the 3 amino acids which contain a hydroxyl group on them. Many proteins, they, their activity is regulated by phosphorylation or dephosphorylation. In order to attach a phosphate group, a hydroxyl group is needed. So these are the 3 amino acids, serine, threonine, and tyrosine, which contains a hydroxyl group on it. And these are the amino acids which can undergo phosphorylation. The next one is asparagine. Asparagine amino acid is an amino derivative of the amino acid aspartic acid. So it contains a 2-carbon chain, CH2 COOH. If it will be COOH, then it will become amino acid aspartic acid. When this aspartic acid, the carboxyl group, undergoes condensation with this amino group, so it becomes an amide bond. So asparagine is an amide derivative of the amino acid aspartic acid. It contains a 2-carbon chain onto which there is an amide bond is present. So this is called amino acid asparagine. The amino acid glutamine, in a similar fashion, is an amide derivative of glutamic acid. Glutamic acid contains a 3-carbon chain and it contains a carboxyl group on it. And when this carboxyl undergoes uh, condensation with the amino group, so it forms an amide bond here. So this amide bond is containing amino acid that is called as glutamine. So glutamine is an amide derivative of amino acid glutamic acid. We will look at the structure of aspartic and glutamic acids after a while. The next amino acid shown here is cysteine. The amino acid cysteine, it contains uh, a sulfhydryl group on it, SH group. So again, if you look at the structure, this is the basic structure of an amino acid, and if you put a methyl group on the side chain, that would be alanine. So onto this alanine, if you take away one of the hydrogens of the methyl group and you put this SH group here, so this will become the amino acid cysteine. This SH group is called as the sulfhydryl group. Cysteine is the second amino acid which contains sulfur in it. So previously, on the previous slide, we have seen amino acid methionine, which contains a sulfur, and the other amino acid which contains sulfur atom in it is amino acid cysteine. Cysteine is important because it is the amino acid which contributes to the formation of disulfide bonds. Many proteins, they contain a covalent bond in their structure which is called as the disulfide bond. So if two cysteines are present close in space, their sulfhydryl groups can undergo oxidation and they form a disulfide bond. Disulfide bond is an important structural component of several different types of proteins. We will look at a few more details about the disulfide bonds in the coming slides. And lastly, here are the amino acids which contain charged groups on their side chains. So their side chains are polar as well as charged. The first one here is the amino acid lysine. The amino acid lysine, it contains a 4-carbon chain on it along with an amino group. This amino group is positively charged. This is a quaternary ammonium group. So a 4-carbon chain, CH2, 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 along with an amino. So that makes the amino acid lysine. The next one is arginine. The amino acid arginine contains this group here. This group, NHC, NH2, NH2. This group is called as the guanidino group. The guanidino group is positively charged. So there is a 3-carbon chain, CH2, 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 and to this chain, a guanidino group is attached. This makes the amino acid arginine. Amino acid histidine is also a positively charged amino acid. And again, if we look here, this is the basic structure of an amino acid. If there is a methyl group attached, that will again become alanine. So you just take away one of the hydrogens and put this ring structure. This ring is called as the imidazolium ring. So the imidazolium ring, it's a heterocyclic structure which contains nitrogen atoms in it, and these nitrogen atom here, that is positively charged. So if you have an alanine, you take away one of the protons and you attach an imidazolium ring here, that will become amino acid histidine. The amino acid aspartic acid, it contains acetic acid as its side chain. So acetic acid is CH3COOH. So if you take away one of the hydrogens here and you put it to this alpha carbon, so that will become a 2-carbon side chain containing a carboxyl group on it. So this is acetic acid attached to alpha carbon. So that is called as aspartic acid. The next amino acid is glutamic acid. Glutamic acid contains a 3-carbon chain. So this is propanoic acid. This is carbon number 1, carbon 2, and carbon 3. So this is a propanoic acid which is attached to the alpha carbon, and that makes glutamic acid. So this structural representation was from Voigt and Voigt. And on the next slide, we are going to revisit the structure of these amino acids and quickly talk about the various groups of amino acids now. And this information is shown from Lehninger Principle of Biochemistry. So here you can see, again, these are the same amino acids which are non-polar, and they are all aliphatic. So again, amino acid glycine having a hydrogen atom as its side chain, and in place of hydrogen, if you put a methyl group, that will become alanine. The amino acid proline contains a 3-carbon chain, but this is different because this side chain is also linked to the alpha amino group. This is something unusual which is not found in other amino acids. The amino groups are free. Valine also contains a 3-carbon propyl chain on it. Leucine is also an aliphatic amino acid, and it contains a butyl chain on it containing 4 carbons. Isoleucine is an isomer of leucine, so you just take away this methyl group and you put it on this carbon here, so therefore leucine will be transformed into isoleucine. And the other one is methionine. Methionine contains a sulfur in it, so CH2CH2 SCH3. This slide shows the amino acids which contain aromatic rings in their side chain. So this is phenylalanine. Again, if you would look at the structure till here, if it's a methyl group on the side chain, that is amino acid alanine. And if you put a phenyl group, a benzene attached here, that will become phenylalanine. And to this phenylalanine, if you attach a hydroxyl group, that will become tyrosine. So the structure till here is the same basic structure, and if it is a methyl group here, that will be alanine. So with this alanine, if you put an indole group here, so that will become the amino acid tryptophan. So these are the three amino acids which contain aromatic rings in them. The next slide shows the charged amino acids which contain charges on their side chains. So first one shown here is lysine. Lysine contains a butyl chain on which an amino group is attached. This is arginine, which contains the gonadino group, and this is histidine. So you attach imidazolium ring to alanine and it will become histidine. So these are the three amino acids which contain positive charges on them. These are the two amino acids which contain negative charges. So this is aspartate. The aspartic acid, it contains acetic acid as its side chain, and glutamic acid, it contains propanoic acid as its side chain. Well, if they're represented as COOH, then it is aspartic acid. But if H, the proton, is dissociated, then that is called as aspartate. Similarly, if it is a COOH here, that would be glutamic acid. But if it is deprotonated, its proton is released. That is called as glutamate. These are the amino acids shown here which contain polar but uncharged side chains. So this is amino acid serine. If you just take alanine and put a hydroxyl group on alanine, so that will turn into serine. This is amino acid threonine. It contains ethanol as its side chain. So you see CH3, CH2OH, that will be ethanol. So you just take away one of the hydrogens from here and attach the ethanol molecule to the alpha carbon. So that will become amino acid threonine. This is cysteine. Again, till here, a methyl group, that will be amino acid alanine. You take away one of the hydrogens and put a sulfhydryl group, that will become amino acid cysteine. This is amino acid asparagine. Aspartic acid contains acetic acid as its side chain. So on this acetic acid, if the carboxyl undergoes condensation with amino and forms this amide bond, that is called as asparagine. Similarly, for glutamic acid, there is a propanoic acid, 3-carbon chain containing an acid carboxyl acid on it. So if the carboxyl undergoes condensation with amino and forms an amide bond here, so this will turn into glutamine. So if you would just follow these series of structural resemblance between these amino acids, so you can see that if you would just remember the structure of glycine, which contains a hydrogen on it as a side chain, it would be easy to remember the structure of alanine, which contains a methyl group as a side chain. And if you would remember the structure of alanine, you can simply substitute a few groups, a few atoms, and therefore they would be converted into other amino acids. So in this way, if you make a correlation starting from glycine, alanine, and so on, so it would be much easier for you to remember the structure of these amino acids. This figure is representing the role of cysteine in the formation of disulfide bonds. So you can see here that this is an amino acid cysteine. This is the alpha carbon, the carboxyl group, the amino group, and this is the side chain. The side chain contains a sulfhydryl group on it. This is another cysteine present in the vicinity. It is present in close proximity in space. Under oxidizing conditions, these hydrogen atoms are removed and the two sulfur atoms will be linked through a covalent bond as represented here. So through oxidation, two cysteines, they will be converted into this structure which is called as cysteine. Cysteine contains two sulfur atoms linked to one another through a covalent bond. This is sometimes called as a disulfide bond. Many proteins, they contain disulfide bonds in them. Not all cysteines present in proteins form disulfides, only those cysteines which are close enough to each other in space so that they can undergo oxidation can form a disulfide bond. So, disulfide bonds are important structural components of several of the important proteins. This process is reversible. Upon oxidation, the cysteines could be converted into cysteine, thereby forming the disulfide bond, and under reducing condition, the hydrogens could be added to these sulfurs and the reaction can go in the reverse direction, thereby breaking the disulfide bonds. Oxidizing agents result in the formation of disulfide bond, whereas reducing agents can reduce the disulfide bond and break it into the constituent cysteines. So disulfide bond formation can take place only under oxidizing conditions. In eukaryotic cells, the environment of the cytoplasm is reducing. Protein synthesis takes place in the cytoplasm by the ribosomes, so disulfide bond formation cannot occur there because the environment of the cytoplasm is reducing. So those proteins where disulfide bonds are to be formed, these proteins are translocated through the signal recognition particle to the lumen of the endoplasmic reticulum. The interior of the ER is oxidizing, and there are enzymes which are important for formation of disulfide bond present there. There's this enzyme called protein disulfide isomerase. This enzyme is important for the correct formation of the disulfide bonds. So the environment of the endoplasmic reticulum is oxidizing. Those proteins where disulfide bonds are to be formed, they must be translocated first into the lumen of the endoplasmic reticulum. Prokaryotes do not contain endoplasmic reticulum. Similar to eukaryotes, the cytoplasm of prokaryotes is also reducing. So disulfide bond formation cannot occur in the cytoplasm of prokaryotes as well. Outside the cell membrane, the space between the cell membrane and the cell wall of prokaryotes, that is called as the periplasmic space. The environment of the periplasm is oxidizing. So those proteins in prokaryotes where disulfide bonds are to be formed, they're first transported to the periplasmic space. And in the periplasm, the formation of disulfide bonds takes place. In case of eukaryotes, the process of disulfide bond formation occurs inside the endoplasmic reticulum. The next important property of these amino acids is their typical absorption spectrum. There are three amino acids which contain aromatic groups in them: tryptophan, tyrosine, and phenylalanine. Out of these three amino acids, tryptophan and tyrosine, these are the amino acids which show a characteristic absorbance at 280 nanometer wavelength. In this graph, you can see absorbance on the y-axis and wavelength on the x-axis. So you can see that these aromatic amino acids, they show a characteristic absorbance at 280 nanometer. So proteins typically show this absorbance at 280 nanometer. Nucleic acids, their absorption maxima is at 260 nanometers. So proteins, they show a typical characteristic absorbance at 280 nanometer wavelength, and that is primarily because of the presence of the aromatic amino acids: phenylalanine, tryptophan, and tyrosine. Among these three aromatic amino acids, tryptophan contributes the most to this absorbance. Many proteins typically contain hundreds of amino acids, whereas other proteins they can contain thousands of amino acids in them as well. So the amino acids are joined to one another through a covalent bond. The covalent bond which joins the amino acids together is called as the peptide bond. Formation of the peptide bond is done by the ribosomes. The amino acids are present as free amino acids in the cytoplasm of a cell. Following the codes of the messenger RNA, the ribosome incorporates those amino acids by the help of transfer RNA. And then these individual amino acids are joined to one another to form the entire protein. The amino acids are joined to each other through this covalent bond, which is called as the peptide bond, as represented in this figure below. So you can see this is one amino acid. This is the alpha carbon of this amino acid, the carboxyl group, amino group, side chain, and the alpha hydrogen. This is the second amino acid. So you can see this is the alpha carbon, carboxyl group, amino group, side chain. So the carboxyl group of one amino acid undergoes a condensation reaction with the amino group of the next amino acid. Therefore, this car carboxyl group will be covalently attached to the amino group. When carboxyl and amino undergo condensation, the resulting bond is called as an amide bond. So the peptide bond, as represented here by means of this red line, is the chemical nature is an amide bond. So this is amino acid number 1, which is linked through this peptide bond to amino acid number 2 here. And now the carboxyl of amino acid number 2 is free. So this carboxyl can further interact with a third amino acid and therefore would result in formation of a second peptide bond here. In this manner, the amino acids they are joined by the ribosome one by one through this peptide bond. The chemical nature of the peptide bond is that it is an amide bond formed by the condensation between a carboxyl group and an amino group of two different amino acids. When two amino acids are joined through a peptide bond, the resulting structure is called as a dipeptide. When three amino acids are linked to each other, that is called as a tripeptide. Four amino acids joined through peptide bonds, that is called as a tetrapeptide, and so on. This structure here shows a pentapeptide. That means there are five amino acids joined to one another through peptide bond. Notice that the number of peptide bonds here is 4. So this is the first amino acid shown here. This is the amino group, the alpha carbon, and the carboxyl group. This is the side chain represented in red color. So the carboxyl group of one amino acid undergoes condensation with the amino group of the next amino acid. So the peptide bond is highlighted here. So this is the second amino acid, amino group, alpha carbon, and carboxyl. This is then joined to the amino group of the third amino acid. And this is the third amino acid till here. And the carboxyl of third amino acid is then joined to the amino of the fourth. And here, this is the carboxyl of the fourth joined to the amino of the fifth amino acid. So this is a pentapeptide containing 5 amino acids linked through peptide bonds. Another important aspect of amino acids is their acid-base properties. Amino acids, as we know that they contain amino group and carboxyl group. The carboxyl group, it can dissociate and release a proton, thereby converted into carboxylate ion, which is negatively charged. In the amino groups, the nitrogen atom of amino group, so the proteins, and because of the presence of these charge groups on their constituent amino acids, they contain charges. The charge is dependent upon the ionization of the carboxyl and the amino groups, which in turn is dependent upon pH. So at a certain pH, where the number of positive charges become equal to the number of negative charges, so therefore the net charge on the molecule becomes zero, That particular pH is called as the isoelectric point of that amino acid. Similarly, proteins are made up of hundreds of amino acids. At a certain pH, where the net charge of the protein becomes zero, that is called as its isoelectric point. That is, in other words, the number of positive charges become equal to the number of negative charges. This figure shows the titration curve of the simplest amino acid, glycine. Glycine has hydrogen as its side chain. It does not have any dissociable groups on the side chain. Some amino acids, as we have seen earlier, they do contain dissociable groups on their side chains as well. But this is the simplest amino acid. On the y-axis of this graph, you can see pH, and the x-axis describes the dissociated protons. So you can see here that the equilibrium point for the dissociation of carboxyl group, the equilibrium point lies somewhere close to pH 2.3. The equilibrium point for the dissociation of the amino group lies somewhere close to 10. So that means the carboxyl and the carboxylate ion are in equilibrium somewhere close to pH 2, and the quaternary ammonium and the amino group, they are in equilibrium closer to a pH of 10. The midpoint of these two is the isoelectric point. This is a simple amino acid and does not have any dissociable groups in the side chain. So therefore, this is a simple average of the two dissociation points. However, for amino acids which contain ionizable groups on their side chain, this pattern is more complex. Proteins which contain thousands of these dissociable groups on them, the pattern is even more complex. Here in this figure, you can see the dissociation curve for glutamate. Glutamate contains two carboxyl groups, one alpha carboxyl, which is represented here, and the second one is the side chain carboxyl. This is the amino group. In addition to these 20 so-called standard amino acids, there are several derivatives of these amino acids which are important for various physiological functions. For example, hydroxyproline and hydroxylysine, they are important for the biosynthesis of collagen. Collagen is a structural protein. Trimethylysine is another derivative of this amino acid lysine, which is important for fatty acid transport. Carboxyglutamate is a derivative of glutamic acid. It contains an additional carboxyl group on it. It is a substituted amino acid which is found in the blood clotting factors. Acetyllysine is a derivative of the amino acid lysine. Acetylation of lysine is important for regulating the binding of DNA to the histone proteins. So these amino acid derivatives, although they are not the standard amino acids, but they are essential for various important physiological functions. In addition to their role as the building blocks of proteins, amino acids are also important for the physiological synthesis of many bioactive compounds. For example, gamma aminobutyric acid is a derivative of amino acid glutamic acid. It is produced by the decarboxylation of glutamic acid. So this is the alpha carbon, this is the amino group, and this is the side chain of glutamic acid, which contains propanoic acid. The carboxyl group, alpha carboxyl group, through the process of decarboxylation is removed. So the remaining molecule is gamma amino butyric acid. This is a neurotransmitter. The next important molecule is histamine. This is a derivative of amino acid histidine. This is the alpha carbon of histidine. That is the amino group. This is the side chain shown in red color. The carboxyl group is removed through the process of decarboxylation. The resulting molecule is called as histamine. Histamine is important inflammatory mediator. It is produced by different types of immune cells and it acts as a chemical regulator of inflammation. Dopamine is a derivative of amino acid tyrosine, and this is also an important neurotransmitter. Thyroxine, triiodothyroxine, and tetraiodothyroxine. These are iodine containing compounds which are produced by the thyroid gland, and again, they're produced by using tyrosine amino acid as a precursor. They're important for regulating energy metabolism and heat production by the body. Citrulline and ornithine, they are related compounds to arginine and they are important intermediates during the production of urea. Urea is a nitrogenous waste compound which is filtered by the kidney. S-adenosylmethionine is a derivative of amino acid methionine. S-adenosylmethionine acts as a donor of methyl group during several important physiological chemical reactions. So amino acids are not only the building blocks of proteins, but the derivatives of amino acids are important for many physiological functions. So amino acids are joined by the ribosome through peptide bond to form proteins. Proteins, they have several levels of organization. As depicted in this diagram, the primary structure of protein refers to the sequence of amino acids, that is, the order in which amino acids are attached to each other through the peptide bonds. The next level of structure is called as the secondary structure. Secondary structure elements are local structures that how various amino acids interact with each other and they form a local structure that is known as the secondary structure. The helices and the sheets, which you have studied earlier, these are examples of secondary structure elements. Various secondary structure elements, they interact with one another and they give rise to the global architecture of the protein structure. That is called as the tertiary structure of protein. The tertiary structure refers to the entire structure of a polypeptide chain. Those proteins which have only one polypeptide chain, their final level of organization is tertiary structure. However, several proteins, for example hemoglobin, they have more than one protein chains in them. For example, hemoglobin has 4 polypeptide chains in it. So these individual polypeptides, they have their own individual tertiary structures. But when they interact with one another, they give rise to this level of organization, which is called as the quaternary structure. Quaternary structure exists only for oligomeric proteins. Oligomeric proteins are those which contain more than one polypeptide chain subunits in them. We will explore more about protein structure and the levels of organization in the coming lecture.